everyone, let's begin with the next chapter of ophthalmology, that is Physiology of Eye. Now if you are able to see this video properly, that means your eye is doing good. That means your eyeball is stable. What do I mean when I say stable? Stable means your eyeball is having normal intraocular pressure as well as the components of your eyeball are also doing fine. Now, how are these physiological activities which are involved in the normal functioning of your eye maintained? It is maintained by the most important thing, that is your intraocular pressure. Now, if I'm saying something like intraocular pressure, that means there is something responsible for this pressure. What is that? That is your intraocular fluid. Now, the intraocular fluid is your aqueous humor. This aqueous humor is responsible for the nourishment and nutrition of various tissues of the eye as well as your intraocular pressure. So let us begin our discussion with the four things, which is nature and formation of intraocular fluid, how this fluid is circulated, how this fluid, that is your aqueous humor, is responsible for your intraocular pressure, and how is this fluid also helping in the metabolism of the ocular tissues, may it be the vascularized tissues like the uveal tissue or the non-vascularized tissues, that is your lens and the cornea. So firstly, let us begin with the nature and formation of intraocular fluid. Now, in body, if I talk in general, if there is an exchange going to happen between two things, that is your tissue and the capillaries, then there must be a barrier, right? There must be a two-way communication for transference. Similar is the case with the eyeball. There is a blood-ocular barrier which is involved in the fluid transfer. Now posteriorly, this blood-ocular barrier that is formed by three things. That is your, yeah, you guessed it right, your retinal capillaries. Then comes your RPE, that is your retinal pigmented epithelium, and lastly your Bruch's membrane. Bruch's membrane is simply the layer between your choroid and the retinal pigmented epithelium. On the anterior side, it is formed by the uveal capillaries. Uveal, that is your iris capillaries, fine. And in the middle side, that is UB, it is formed by your ciliary, epithelium and capillaries. Ciliary epithelium and capillaries, which will drain your fluid into the posterior chamber of the eye. So this was about the blood ocular barrier. Let us talk about the intraocular fluid. When I say intraocular fluid, the main thing is the normal aqueous humor. But there is also the vitreous, as you must be knowing. So let us quickly finish it off first. Fine. So the vitreous humor or the vitreous, it is a stagnant gel. So it is a stagnant gel. It is mesenchymal in origin. It is mesenchymal in origin, formed by the ciliary epithelium. It is composed of type 2 collagen as well as hyaluronic acid. And it consists of many cells. Mainly these cells are phagocytes, which will phagocytize the cell debris, and also hyalocytes, which are lining your vitreous humor. And it is composed 98 to 99% of it is composed by water. Then coming to your normal aqueous. So the aqueous, it is formed by the ciliary epithelial cells. Your aqueous humor is formed by your ciliary epithelial cells. And do you know, your aqueous is nothing but you can say it is formed from the plasma by different mechanisms. Now what are these mechanisms? This is ultrafiltration, diffusion and secretion of plasma through the ciliary epithelial cells. This diagram, these are your ciliary epithelial cells. They are adherent with each other via tight junctions through which secretions happen. Fine. So ultrafiltration, diffusion and secretion of plasma, secretion from the plasma by ciliary epithelial cells will result in the formation of your aqueous humor. Now this aqueous humor, it is a transparent fluid. It is colorless. It has low refractive index. It has low refractive index, and you know it is actually having more lactic acid and other things like vitamin C or ascorbic acid and pyruvate. It is more in amount even than the blood. Why is it so? We learn later on. I'll tell you in short that it is due to the anaerobic glycolysis happening in the lens. And this lactic acid, which is formed as an end product of this anaerobic glycolysis, it will ultimately accumulate in this aqueous humor. Fine, I'll tell you this later on also. So this was about your vitreous and your aqueous. You need to remember, this is important, that the vitreous is a stagnant gel, whereas your aqueous, it is a transparent fluid. Fine. So this was about the formation of intraocular fluid. Now once it is formed, how is this aqueous humor circulated? Now why is this necessary? It is necessary for both metabolic purposes and to regulate the intraocular pressure. Do you know the non-vascular tissues, that is your cornea and lens, they depend on the aqueous humor for their nutrition because they are not having any blood supply of their own, right? So once this is formed, how is it drained? From the ciliary bodies, it will form the aqueous. Fine. Then it will accumulate in the posterior chamber. If I say this is your ciliary bodies, then this one is your ciliary body. It will drain the aqueous in the posterior chamber through this one is your lens. This one is your zonules. Fine. This one is your iris, and this one is your pupil. So once it is formed in the posterior chamber, it will go via the pupil. It will go via the pupil into the anterior chamber. Okay, it will go via the from the posterior chamber via the pupil. It will go into the anterior chamber, and from the anterior chamber it will go at the angle of the cornea. At the angle of the cornea, it will go into the trabecular meshwork. Just follow my arrow. It will go into the trabecular meshwork, which is a connective tissue at the junction of the cornea and the sclera. From the trabecular meshwork, it will go into the Schlemm's canal, and from the Schlemm's canal, it will go into various collector channels and aqueous veins, and finally, this aqueous humor will drain into the episcleral veins. Fine. So this was the primary drainage. I'll just repeat it in short once again. The ciliary bodies, they will form the aqueous humor and they will drain it into the posterior chamber. From the posterior chamber, this fluid will go via the pupil. Pupil is just an opening between the iris. It will go via the pupil into the anterior chamber. In the anterior chamber, it is your anterior chamber, and from the anterior chamber, it will drain at the trabecular meshwork, Schlemm's canal, collector channels and aqueous veins, and finally in the episcleral veins, which will go in the systemic blood. Fine. So that was the primary drainage. Apart from this, it has got a uveoscleral outflow. Uveo means the uveal tissue, that is your vascular tissue, that is your iris and choroid and ciliary body wala part. Fine. So this also allows aqueous through the ciliary body into the choroid and suprachoroid and into the episcleral tissue. So this is a minor pathway, or we can say an accessory exit outflow. Fine, that is through the uveoscleral outflow. This was about the circulation of aqueous humor. Next, we'll learn about the intraocular pressure. So the normal intraocular pressure is around 10 to 20 millimeter of mercury. If the intraocular pressure exceeds or it goes beyond 20 or 21, this is a condition you might be knowing, that is known as glaucoma. So how is this intraocular pressure maintained? It is like a balance between the production and the drainage. Production by the ciliary body and drainage via the canal of Schlemm. Fine, or the Schlemm's canal. So it is a balance which is maintaining the intraocular pressure. Other factors, like they can exert few temporary effects. What are these? First is your variation in the hydrostatic pressure in the capillary and an increase in permeability of the capillaries. I told you that the retinal capillaries, they are relatively impermeable. Fine, impermeable to large molecules. Now, if the permeability of the capillaries will increase, then large molecules will easily enter the eyeball, right? So this condition or this aqueous humor thus formed is known as plasmoid aqueous. This is having proteins, fine? Large molecules due to increased capillary permeability. This aqueous is known as plasmoid aqueous. Then comes osmotic pressure of the blood. As you know that this aqueous is nothing but ultimately it is formed by filtration and you can say diffusion, secretion of the blood. So if there are changes in the osmotic pressure of the blood, it is ultimately going to reflect in your intraocular pressure and your aqueous. Fine. Then coming to volumetric changes. Now volumetric means the fluid which are present in the eyeball, may it be aqueous or the vitreous. If something happens or if there is an increase in its volume, then it will ultimately really, reflect into the increase in intraocular pressure. Example is your vitreous hemorrhage, in which there is leakage of blood vessels into the vitreous, leading to blood in the vitreous humor, which will result in raised intraocular pressure. And lastly comes your blockage of the circulation of aqueous. I told you that the ciliary bodies that will drain your aqueous into the posterior chamber, right? Then the posterior chamber via the pupil, it will go into the anterior chamber. Fine. So if there is a blockage at the pupil or at the angle of cornea, see here, if there is a blockage over here or if there is a blockage over here, it is not clear in this diagram, the pupil or the canal. So that may result in increased pressure. So that was about raised intraocular pressure. Normal pressure is 10 to 20 millimeter of mercury. Fine. Then comes metabolism of ocular tissues. Now, what do you mean by metabolism of ocular tissues? Now, there are two types of tissues in the eye. One which are vascularized, that is having their own blood supply, which are known as the uveal tissues. So, they will not differ from the general metabolism which occurs in the other tissues of our body. Fine. So, that will metabolize in the same way as the other tissues do. Coming to the non-vascularized tissues of the eye, that is they are avascular, not having their own blood supply. So how will they metabolize? How will they gain nourishment? Let us learn that. It is very important to learn this. Fine. So the first and foremost thing important over here is their main source of nutrition is carbohydrates, and these carbohydrates are utilized by phosphorylation and auto-oxidative mechanisms. Now, your non-vascularized tissues are your cornea and your lens. Let us learn about the cornea first. The cornea, it has low energy requirements. Then, why it is necessary to metabolize or to nourish cornea? Because it needs to replace its tissues. Fine. And last thing is transparency. Cornea needs to be transparent. Fine. It needs to maintain its transparency, and this depends essentially on its state of relative dehydration. The cornea must be dehydrated. And how does it maintain its dehydrated state? Via fluid exchange. Fine. So it will continuously exchange its fluid. Now, what are the sources of nutrition or metabolism for cornea? First thing, oxygen directly from the air. As you know, the cornea is the outermost layer of your eyeball. Fine. So it is in direct contact with the atmosphere, and thus it can gain oxygen directly from the air. Then comes solutes from the perilimbal capillaries surrounding your limbus. And lastly, the important thing is your aqueous humor. So these are the three sources, and major pathway of utilization is aerobic glycolysis. The cornea can easily utilize oxygen to perform aerobic glycolysis. Coming to your lens, so the lens will derive nourishment entirely from the aqueous humor, by capsular and subcapsular epithelium, Cornea did the same thing by corneal epithelium. Fine. Lens is doing it by capsular and subcapsular epithelium. And what are the methods or modes by which it can use it? It is using it primarily by anaerobic glycolysis, because the lens is not in direct contact with oxygen. It can only utilize the oxygen which is present in dissolved form in the aqueous humor. So it mainly undergoes anaerobic glycolysis and it will generate ATP. 70% of ATP is generated by anaerobic glycolysis, and it can also perform TCA cycle and HMP shunt. TCA cycle is aerobic pathway, right? It can utilize some amount of oxygen via the dissolved oxygen in aqueous humor. So that will lead and give up to 25% of ATP. And NADPH can also be a source of energy via the HMP shunt. So these are the important thing in lens. And the last important point is the metabolic activity of the lens is largely confined to the cortex. The older nucleus is relatively inert. So as you all know, if this is the lens, then the outermost layers are more active. That is your cortex, fine. Cortex, and the inner layers are known as the nucleus, may it be embryonic, fetal or infantile nucleus, fine. So this was about the cornea and the lens and the metabolism of ocular tissues. Lastly is the major factor responsible for transparency of the ocular media is their avascularity, the cornea and the lens. So they are avascular structures, and thus they help in maintaining their transparency. And if someone asks you that what are the refractive media of the eye, so these are the things you should know. The tear film, that is your tears, that are your tears. Then comes your cornea. Cornea is the main refractive medium of eye. Then comes your aqueous humor, helps in maintaining intraocular pressure. Then comes your crystalline lens. Lens also help in focusing mechanism, fine. And lastly is your vitreous humor, which performs phagocytosis. So this was all about the physiology of eye. If you like this video, please do like and share. If you have any comments, doubts or feedbacks, do let me know in the comment section. Thank you all.